The first thing we need to know is what is the difference between eukaryotic and a prokaryotic cell. Now, prokaryotic cell is a bacteria and eukaryotic cells are plants and animals. Now, if you see in this diagram, there is a eukaryotic cell and a prokaryotic cell. The first basic difference between the two is the presence of this structure called the nucleus. So nucleus is present in a eukaryotic cell, but in a prokaryotic cell, the nucleus is absent. Apart from the nucleus, eukaryotic cell, you can see they have those organelles which have membrane around them. So all membrane-bound organelles are present in a eukaryote, example mitochondria, but these membrane-bound organelles are absent from a prokaryotic cell. The next difference is, since the nucleus is absent, in the eukaryotic cell, the DNA is enclosed in the nucleus, but in a prokaryotic cell, it lies naked in the cytoplasm as it has no nucleus. The eukaryotic cell are mostly multicellular, but the prokaryotic cell are mostly unicellular. The eukaryotic cell DNA is linear, but prokaryotic cell DNA is circular. Both eukaryotic and prokaryotic cell has ribosomes, but in a eukaryotic cell, the ribosomes are big. In a prokaryotic cell, they are small. Eukaryotic cells are multicellular big cells, whereas prokaryotic cells are small unicellular cells. Example is a plant cell and an animal cell of a eukaryote, and for a prokaryote, there's a bacterial cell. So I hope you're able to write these differences, because in the exam, they can ask you to list, any two differences or any two similarities between a eukaryotic cell and prokaryotic cell. So make sure you understand these points. Next is, let us look at in detail the structure of an animal cell. Now the diagram here shows you the animal cell, and you can see there's a round big nucleus. The jelly-like fluid that fills the cell is the cytoplasm. The outer layer that surrounds the cell is the cell membrane. And then in these structures, on them and freely, you have ribosomes. And then you have these pink structures called the mitochondria. Now, you should know these names and the functions of each, as these are found both in plants and an animal cell. So the first is the nucleus. It is the brain of the cell, because it controls all the activities. It controls what is happening inside the cell, and it also has your genetic information as it contains the DNA. So you should know all these three functions of the nucleus. Next is the ribosomes. Ribosomes are the site for protein synthesis. Now the cell needs proteins for its functioning, and enzymes are also protein. So ribosomes, they make proteins and the enzymes which are required by the cell. Next is the cytoplasm. The cytoplasm is like the jelly-like fluid which fills the entire cavity of the cell. It is a very important part because it has all the enzymes for the chemical reactions of the cell. So majority of the chemical reactions of the cell takes place in cytoplasm. Next is the cell membrane, as it's the outer layer. So as it's the outer layer, it is controlling what is going in and out of the cell. And mitochondria is very important. It is a powerhouse of the cell. Powerhouse means it provides the cell with the energy, as it is the site where aerobic respiration takes place. Okay, so I hope you know the function of each of these organelles. Okay, and you should be able to label them. If any cell is given to you, you should be able to spot where's the nucleus, where's the cell membrane, where's the mitochondria. Next is the plant cell. Now the plant cell has all these organelles which we have just discussed in the previous slide, except it has 3 extra organelles which are just found in a plant cell. This is a big, large permanent vacuole. These green structures called the chloroplast, and outside the cell membrane there's an extra layer called the cell wall. Now, the permanent vacuole, you should remember the function of each one of them. The permanent vacuole is filled with the cell sap, and it gives rigidity to the cell and makes the cell turgid. The cell wall is the outer layer, and it is made up of cellulose. You should remember the cell wall of the plant cell is cellulosic. It is a layer outside the cell membrane, and it supports the plant and maintains its shape. The chloroplast is the site of photosynthesis, because it contains a green pigment called chlorophyll, which absorbs sunlight and converts the light energy into chemical energy, making food. Okay, so you should know the difference between an animal cell and a plant cell. So this is a table that summarizes the difference in both plants and similarities. So in the exam, they can ask you to, give 2 similarities or 2 differences. So you should know both similarities and differences in both of these cells. So both are eukaryotic. The nucleus is present in both of them. Cell membrane is present in both of them. Mitochondria is also present in both of them. Ribosome is also present in both of them. And cytoplasm is also present in both of them. But cell wall, vacuole, and chloroplast is present only in the plant cell. So it is absent in an animal cell and it is present in in a plant cell. Okay, so this difference is very, very important. Next is the bacterial cell. You should know a typical prokaryotic cell, which is a bacterial cell. Make sure you can label it. So you can see the tail-like structure, which is the flagellum, which helps the bacteria to move. Then it has a cell wall and the cell membrane. But the cell wall is not made up of cellulose. You have to remember here, it is made up of peptidoglycan. Then outside the cell wall, you see a slimy layer, which is a capsule, and this capsule protects the bacteria. And you also see some hair-like structures outside this cell wall. These appendages helps the bacteria to reproduce. Okay. Then, since it has no nucleus, the DNA is circular and it is found naked in the cytoplasm, and it has some extra chromosomal material. You can see here they have plasmid. Now, plasmids are the extra chromosomal materials which are found in the bacteria. So you can see these extra stretches of DNA. These are plasmid, and they are in the form of small rings, and they give special properties to bacteria like antibiotic resistance. Okay, so I hope these all organelles, this labeling and their functions are clear to you. Okay, so now this is similarities and the differences between the bacteria, plant, and an animal cell. Bacteria is prokaryotic, plant and animals are eukaryotic. The nucleus is absent in bacteria but present in both plants and animal cell. Cell wall is present in bacteria but non-cellulosic. Remember, in a plant cell it is cellulosic, in animal cell it is completely absent. Cell membrane is present in all. In bacteria, ribosomes are smaller, but in plant and animal cell they're bigger. DNA is circular in bacteria but linear in plant and animal cell. The genetic material in bacteria is naked, found in the cytoplasm. In plant, it is in the nucleus inside the chromosome, and same in animals. The chloroplast are absent in the bacteria but they are present in the plant and animal cell, and the vacuoles are small in bacteria but in the plant and animal cell they are absent in the animal cell but present in the plant cell. Okay, so I hope the similarities between bacteria, plant, and animal cell and the differences, they are clear to you. Okay, so next, in your syllabus, there's a topic on order of magnitude. Now, what is order of magnitude? They'll give you the size of the two cells, and then they'll ask you what is the order of magnitude of the two cells. So what you need to do is, you need to divide the size of the two cells, okay? And that will give you an order of the magnitude. So for example, if one cell is, let's say, the 10 meter, and the other cell is, let's say, 100 meters, right? So you divide the two size, so you will get the answer as 10. So it means that one cell is in the order of magnitude 10 than the other. Okay, so for that, it is very important the units are same. So in the exam, they can give you one in meters, another in nanometer, one is in micrometer, another in millimeter. So you should remember this conversion. So this conversion is simple. It comes as a chart: killing metal Milo, Mickey nano pictures. Okay, so killing metal Milo, Mickey nano pictures, it's kilometers, meters, millimeters, micrometers, nanometers, and picometers, and all are at the level of 10 to the power 3. So kilometers to meters times by 1,000, times by 1,000, times by 1,000. And when you go reverse, you need to divide by 1,000. So for example, if you need to convert the nanometer into meter, so you're going from here to here. So you will divide it by 10 to the power 9. Okay, so 1 nanometer is 1 times 10 power minus 9 meters. So you can remember this table, and this is a mnemonic to remember it. So if it is 10 nanometer to meters, so from nanometer to meter, what do? You divide it by 10 to the power 9. So that becomes 10 power minus 8 meters. And micrometers to meters, so micrometers to meters is 1, 2. 2 bumps, so it means divide by 10 to the power 6. So that gives the answer 10 power minus 5 meters. So it's very important. You know how you need to convert. Make sure both the units are same and then divide the two. Okay. Now, the next thing we need to do is, what are the specialized animal cells? Now, what are specialized animal cells? Now, specialized animal cells are those cells which have some extra features that allow them to perform the specific functions. Okay. Now, What do you need to do for each of these cell type? You need to know what are the functions of each of these cell type, and depending on the functions, what structural characteristics they have. So, for example, a nerve cell. What is the function of the nerve cell? To send electrical impulses around the body. Now, this is the structure of the nerve cell. And in this nerve cell, what we have is that the nerve cells have dendrite. Now, what is this dendrite? These dendrites are the hair-like structures that receives the impulses. Then it has a long stalk called the axon. This axon transmits the nerve impulses, and at the end you have the synapse. Now, what is a synapse? They transmit the nerve impulses from one neuron to another. So the structure goes like this. It has a nucleus, hair-like structure, and a long stalk, and at the end it has the synapse. Next is the muscle cell. The function of the muscle cell is to contract and bring about the movement of different parts of the body, and for that they are made up of the special fibers which helps them to contract and relax. They contain special proteins that allow them to contract and relax. They have loads of mitochondria which provides them energy to contract, and they have special storage carbohydrates called glycogen which acts as a fuel for the muscles. Okay, so for both of them, you should know what is the function and what features do they have. Next is the sperm cell. Now, if you look at the sperm cell, it looks like this. At the top, there's a head which has an acrosome, and this acrosome contains hydrolytic enzyme which breaks the egg cell wall and penetrate inside the egg to fuse with the egg nucleus. The middle piece has mitochondria. The mitochondria provides the energy to swim. The nucleus contains the genetic information, and the flagella is a tail-like structure which helps it to swim to large distances. So I hope this function, and you should be able to label this like what is an acrosome with its function. Okay, the flagella with its function. So you should know the different components of a cell along with their functions. Okay, so now as we have finished the specialized animal cell, in the same way we have the specialized plant cell. Now, what are the specialized plant cell? Again, these are those cells which have some extra features that allows them to perform special functions. Now, again, to write about these, you should first know their function. So root hair cells is a cell which act as an exchange surface and absorb water and minerals. Now, for the absorption of water and minerals, it needs a large surface area. So the root hairs are the extensions which provides a large surface area for the absorption of water and minerals. Then it has a large permanent vacuole which supports the cell and speeds the movement of water by osmosis. At times, it's not just the diffusion through which the water and minerals move. The active transport also happens. So for that, there's a mitochondria inside the root hair cell, which provides the energy for active transport. It has a cell wall, and it is just one cell thick so that the substances can easily diffuse and the diffusion pathway is reduced. On the other hand, there's a xylem cell. What is the function of the xylem cell? It transports water and minerals from roots to all parts of the plant. Now, since it has to travel water to large distances, it has to form a long continuous column. Now, the cells of the xylems are dead so that they can form a long continuous column which can transport water to large distance. And then inside, you can see the lignin deposit. The lignin deposit makes it waterproof and makes the xylem column strong and strengthen the xylem vessel. The phloem cell is again a transporting tissue, but its function is to transport food from the leaf to all parts of the plant. Now, the phloem has lost majority of the cell content so that it can have more room for the food. The cells at the periphery, so it is in the form of the sieve tube, and the cells which are at the end of the sieve tube, they are broken to form a sieve plate so that the food can easily percolate. Okay, and since the sieve tubes have lost majority of their cell contents, they are supported by a special cell called the companion cell. It supports the phloem cell and provides the cell whatever they need. Especially, they are rich in mitochondria, which makes a lot of energy for the sieve plates and sieve tubes so that they can transport food. The last is the photosynthetic cell. The function of the photosynthetic cell is to prepare food by photosynthesis. So it has a large vacuole which helps in the movement of water by osmosis. It having loads of chloroplast that helps it to trap light for photosynthesis. Also, these cells lie flat onto the leaf surface near to that so that they can easily absorb water, carbon dioxide for quick photosynthesis. Okay, so I hope these special plants and animal cells are clear to you. Now the question comes in, the cells we cannot see by the naked eye. So we take the help of the microscopes. So microscopes are the devices that use to see the cells which we cannot see by our naked eye. And there are two properties of the microscopes which are important, and you should know the definition of them. First is the magnification. Magnification is the property of the microscope to enlarge the object, and resolution is the property of the microscope to distinguish between two closely placed objects. Now, magnification is calculated by the image size over an object size. Now, this is an important question. In the exam, they can give you an image size and they can ask you to find the object size if the magnification is given, or the image size and object size is given to you and ask you the magnification. Or to make it a little complicated, they can give you a cell and they can say that this diameter is 2 micrometer in reality, calculate the magnification. So in that case, what you'll do, you'll take your ruler, measure this actual length, that will be the image size. The object size, they have already given you 2 micrometer. Make sure you make the unit same by the chart that I have just discussed. I'll just put it again. And then you find the magnification by doing the image size over the object size. Okay. So the table that is given before helps you to convert the different units into a single unit. And remember, for the magnification, both image size and object size should be of the same unit because magnification is the ratio. Okay, so now there are two types of microscope. First, we had the light microscopes. These are the microscope which you see in your school laboratory. They are easy to handle, small and compact, does not require much expertise to handle, and they just have either a mirror at the bottom or a bulb, as it uses the beam of light to focus on the object. Whereas electron microscope, it is not easy to handle, it is big and non-portable, requires proper training, it uses beam of electron to focus on the object. Okay. Now, when we use a light microscope, we cannot see the greater subcellular details because the light microscope has a very low resolving and the magnifying power. The resolving power is 0.2 micrometer and magnifying power is just between 1000 to 1500. Whereas for electron microscope, it has a greater resolving power around 0.5 nanometer and the magnifying power is around 100,000 times. So it is the electron microscope that have enabled us to see a proper subcellular structures like mitochondria, ribosomes, and chloroplast. But for the light microscopes, you can view the live samples and you do not require special preparation of the samples. But for the electron microscopes, the samples have to be dead, and we require some special sample preparations. Light microscope, we can just see the color images. We can stain the sample and see the color images. Whereas electron microscope, it can form 2D and 3D, but the images are black and white. Okay, so I hope this difference between the light microscope and the electron microscope is crystal clear to you. Okay, so make sure you can write the similarities and the differences between the two. Okay, so now you should be able to know the difference between a plant cell and an animal cell, the bacterial cell, the light microscope, and the electron microscope. Well clear with this topic. Now what you need to do next is check the specification, make sure the specification is clear to you, and then you need to do the exam question on this topic. If there's still anything that you do not understand, leave a comment below and we'll reply you as soon as possible. Or you can come to our website where we have set up a 24/7 chat support till your exam. You can come anytime, ask any questions, and you can get instant reply. If you like this video, then do subscribe to our channel and do not forget to like, comment, and share this video. If there's any specific topic you want me to make the next video on, then also do leave a comment below and I'll make sure I put that up before your exam. Okay, so I'll see you next in the next video of the next section. Till then, happy revising.